Hello and welcome to the second video on enzymes. You already watched a video introducing you to chemical reactions and also just the basics of enzymes. And remember, enzymes are a type of protein, and basically proteins have a lot of different jobs, but enzymes help speed up chemical reactions. Okay, so just remember that chemical reactions do several things. We can have two molecules come together and form a bond or form many bonds to make one molecule. We can have one molecule actually break bonds or multiple bonds and make two or more products. And then finally we can have two molecules undergo changes, not necessarily form bonds together, but they can undergo changes to create two different products. And so remember, the two types of reactions that we get in chemical reactions is we can have energy absorbing reactions where we actually absorb more energy than is let out. And then we have energy releasing reactions, which means that we basically release more energy than is absorbed. So here is the absorbing energy to get that reaction going, but then we release quite a bit of energy here, and as you can see, from this line to this line is the amount of energy that is released, and more released than absorbed altogether. So this reaction is actually called an exergonic reaction. And this reaction over here, when we absorb more energy than we release, is called endergonic reaction. All right, so chemical reactions with enzyme, here is looking at that same graph but with an enzyme. And so here we have our reactants, here we have our products. The blue line represents the chemical reaction without the enzyme. Notice how much energy from here to here it took to go through that reaction without the enzyme. And if you look at the red line, we see the reaction with an enzyme. And so what we see is less activation energy with the enzyme. It's a lot less. It goes from this line to this line instead of all the way up here. All right, and remember, we get the same reactants and we get the same products when we do a reaction with an enzyme. We're still going to have the same products, it just happened a lot faster. Now, I want to talk to you about the enzyme substrate complex. For every single enzyme, there's a substrate. And we talked about the lock and key method. That substrate is basically a molecule or several molecules that will fit into what is called the active site. And the active site is basically the site where that substrate fits. And it's like a lock and key mechanism. That substrate is actually specific to the enzyme's active site. And what that means is if this is a molecule, let's say an example of a substrate would be hydrogen peroxide. And this is an example of an enzyme called catalase. And basically, hydrogen peroxide will fit into the active site of catalase. Hydrogen peroxide will not fit into any other enzyme's active site because it won't fit. It's a very specific lock and key type of mechanism. And furthermore, another substrate won't fit into catalase's active site. So it's very specific, and every chemical reaction has a very specific enzyme with a very specific substrate. You can think of the substrate as the reactant, and so usually the substrate is what we start with. It fits into the active site, and then what we get at the end are products. Now, the other thing I want to mention is when we have the substrates and the enzyme together, we actually call that the enzyme substrate complex. And so here's an example where we have water and carbon dioxide. They come together and they fit into the active site of this enzyme. And when they do, many chemical changes take place inside this active site where, in this case, these molecules are actually bound together and they're converted into products and then they're released. And so carbonic acid is our product, but I want you to make sure you understand that the enzyme is the same. It doesn't convert to anything. It doesn't change. It actually stays the same shape. Now, there might be some very, very, very subtle changes to the active site, especially when we have water and carbon dioxide coming in, but those changes will come right back around. And so that enzyme is actually good to go again. As soon as it sees another substrate, which would be water and carbon dioxide for this particular enzyme, it's ready to go again. And so it'll go through this cycle over and over and over as fast as it can. And it'll be ready for those substrates as soon as it lets go of the products that it makes. So one thing to keep in mind is that that enzyme is not used up. It's not changed, and it's there for substrates to come into the active site, and then it'll change it in the product. Now the reactants, which are substrates, those actually will get used up. If I put water and carbon dioxide and this carbonic anhydrase in a vial, the water will eventually get used up, and so will the carbon dioxide, and what we'll end up producing is carbonic acid. And so these amounts will change as the reaction goes forward, but the amount of enzyme doesn't ever change. Okay, now what can affect the function of enzymes? Well, there's many things that will affect the function of enzymes, and two examples is temperature and pH. For example, here we have the effect of temperature on a reaction, and what we're seeing here are three reactions kept at different temperatures. There's the same reaction, same substrates and same enzymes are being used, and we're measuring the product that's made. And so here we have 25 degrees Celsius, here we have 35 degrees Celsius, and here is 45. And what you're seeing here is a difference in product. And so there is an optimal temperature that this enzyme works at. Can you guess which temperature that is? It is. It's the 35 degree temperature Celsius. And so this particular enzyme, although we don't know what that is, it doesn't say on this graph, actually works best at 35 degrees Celsius. Same thing with pH. We actually have enzymes in our stomach that work optimally in our stomach. If those enzymes were to get out of our stomach and, I don't know, say work in our arm, they wouldn't work right because the pH in our stomach is very, very acidic, whereas the pH in our arm muscles are not very acidic. It's more closer to a more neutral level, which would be around 7. And so for this graph, we're seeing pepsin is basically one of the enzymes in our stomach. And trypsin is another enzyme that's actually outside of our stomach. And this is the relative activity of these enzymes. And so pepsin works best right at about 3 pH. And trypsin works best right at about pH of 8. And so these are two different enzymes that basically work best depending on their specific environment. And they work best at different pH levels. And so just keep in mind that basically temperature and pH can affect enzymes. We're going to be doing a lab in class this week where we explore that. All right, well, I hope that helps. Until next time, thanks.